My job here is to introduce our keynote speaker for this morning. This is Rusty Gage. So most of you probably know him. Rusty is very well known in the field of neurostem cells, was a pioneer on showing brain neurogenesis even in the adult brain, which is something fascinating. Still lots to be discovered there. But to me, Rusty is a big mentor. So I came to his lab as a postdoc from South of America, just landed in Rusty's lab. And I learned lots of things. I mean, I learned about stem cell biology. I learned about neuroscience. But I think the most important thing was to learn this intuition. So Rusty was never afraid of new ideas or pushing the boundaries of knowledge. So even the craziest project, he was encouraging me to go there. And I would like to introduce him with a quote from Rainer Maria Rilke. This is a German poet. And Rilke said that life is attached to the difficulties. And all the time when I came with a big problem, Rusty said, if it's difficult, it's another reason to be done. And with that, I would like to introduce Rusty Gage. Thank you, everybody.
All right. Thanks very much, Allison and Kat. Yeah, it's great to be here. Congratulations on all your successes. So today, I'm going to focus on one aspect of the work that we do in the lab. In particular, I'm going to focus on this one aspect of stem cell biology that has emerged across disciplines, not just in neuroscience, but other areas. And focusing on organoids, and I'll go into a little in-depth with that. And so when we're studying and trying to understand how the brain works and how disease initiates and progresses, the most important thing we have to have is some model that we can do experiments with. We're experimental biologists. We need to have model systems to understand mechanisms. And a pitch for that really is that to be able to develop medicines, as Kat was saying, is good science, but I think we need to have basic mechanisms of how disease progresses at a molecular level. And you really need models for that. In the last decade or so, the emergence of induced pluripotent cells has come and demonstrated a terrific utility. And this is by taking blood or skin from patients and differentiating them into induced pluripotent cells or stem cells. And these embryonic-like stem cells can be differentiated into a lot of different cell types. That is progress. It's going on. It's working well. What was missing was some sort of three-dimensional context. And a little over a decade ago now, a variety of people had initiated actually in the eye, developing a cup for the eye. These organoids were developed, and they've emerged and become very useful as a culture system for three-dimensional aspects. And of course, animal models are continue to be important for what's going on. When we started using organoids about 15 years ago, or started working with them, one issue emerged for us that was difficult to get around, and that is that once the cells reach a certain density, they have a necrotic core that evolves because there's a density gradient where molecules can't get to the core. And Arne Kriegstein and his colleagues sort of came up with this description. An underlying mechanism may have a solution to that, or not a solution to that, but a description of why it happens. And then Knoblish himself came up with a computational way of screening out the cells so that you could look specifically at the healthy cells. And so lots of different ways of approaching it. And I think that the in vitro organoid situation now has stabilized a bit and is working well. One of the things that was missing for us was this idea of vascularization. And so based on some work that I did, I don't want to say this, 40 years ago, I imagined that you could take these three-dimensional structures and transplant them and as a xenograft into an immunocompromised mouse, so they have a humanized immune system or challenged system. And in our early studies 40 years ago, we could see fetal tissue being innervated and by the vasculature from the host. So it was a true chimera. And here you can see that we can transplant them, and that this just gives you a little video showing when you inject a red dye into the orbital fissure that you get a flow of blood into the vein that's inside the organoid. And so we've done this, and one of the advantages of this transplantation system is you can put a sort of a glass cover slip over the top of the organoid and then use two-photon imaging to look inside the living human brain, vascularized brain, and begin to discover things that way. So we have, let's see here, we've done this, and here's the situation where we take organoids, we make them, and we use a apoptosis marker tunnel, and here's the same organoid starting at this early time point, and then in vitro there's an increase in cell death, but if you transplant them, you really reduce the amount of cell death. When we look inside, we see that there are actual synapses that are being formed. Pre- and postsynaptic densities are forming against each other within this human space, and we can look at the physiological activity of them. Early on, they have this typical brain, early development brain structure where there's this synchrony that occurs, and then over time, the activity matures in the cells and they become desynchronous and more adult-like. But one of the things we recognized as we were developing these stories is organoids really are neuronal organoids for the most part. They're made of neurons and neural progenitor cells. So now we have endothelial cells and parasites coming in from the host, but those are mouse. They're not human. And this line that you'll see me talking about throughout this talk is trying to make a humanized three-dimensional structure. Well, one of the cells that we're particularly interested in is in this glial lineage, and I'll talk about two different cell types, microglia and astrocytes. In particular, there's been great success recently showing that you can take fibroblasts or iPS cells even and differentiate them directly into microglia. And these microglia retain many of the activities that they have in vivo in an in vitro setting as a monolayer. But they're missing a lot of the genes. And here's a series of studies, two brilliant ones by Ben Barres before he passed away, talking a little bit about these glial cells. But here, these three genes right here that you can't really see in any kind of detail are the mature markers, and they're all missing in these three-dimensional cultures. Chris Glass and his colleagues at here at UCSD obtained microglia from postmortem tissues and from biopsies from children and found out that the fresh dissociated microglia retained this maturity to them, but as soon as they were put in the culture dish, that within hours they de-differentiated and became much more immature. So we had to find how can we retain and study microglia, which are becoming so important for disease. So we thought about the timing of this, and we made what were called epithelial progenitor cells, and these precursors to microglia that can be derived from iPS cells can be incubated in vitro with the organoid. So here's the organoid, and here are these progenitor cells, microglia progenitor cells that are migrating into the organoid. If we wait for 3 months after the incubation of the microglia into the organoid, they now can express these markers that we have been long searching for, which includes the TIS-TM19 and other mature microglia markers. We've done single-cell sequencing on these and showed that over a period of time, from 5 to 8 months, they mature in much the same way as Ben showed in the human adult versus fetal tissue. You see a maturation occurring. So they really can mature into these mature functioning cells. And here's an image of the transplanted organoid, and these are the we label the iPS cells that are from which we make the microglia, and that way we can visualize them and see them in the tissues. Now, one of the things that was a real surprise for us, and this is work done together with Axel Nimmerham, who's a imager at the Salk Institute, and he uses two-photon imaging. One of the unique things about microglia is that, if you think of static images and you think of cells that are sort of sitting in the brain, Well, these microglia are sensing cells. These are the immune cells of the brain, and they sense the environment all around. And they're constantly active under a normal setting, and they're sensing environment. And he had shown this some decade ago in mice, but this is the first time that he saw human microglia behaving in the context of human brain tissue in a transplanted organoid in the mouse. Now, they're actually functional. You can treat the animals with IPSs or any kind of stimulus that would activate an inflammatory response, and they move from these active searching cells into a macrophage. And so they are, not only are they behaving, but they're responding to the environment in the way that you would expect them to behave. Here's another trick that he does. He kills a cell right here. And what happens is that these microglia that are normally sensing the environment, they're secreted nucleotides from the dying cells that attract these processes, and then they phagocyze the cells in there. So this is a little bit different than what happens in the mouse, because in the mouse, the microglia they still sense the death that's occurring in the cell, but they move toward it and they phagocytize it. Whereas humans are so big, these microglia are so large, they send their processes in, maintaining. But they still move about halfway there, but then they can do it. And I will talk about that a little bit more. One of the amazing things this is a demo of how this tool can be used. This is close to Alzheimer's part. This is a study that we did related to autism. There's a form of autism where they have macrocephalia. Their brains are large. And one of the things that people have learned about this, people from human imaging, is that the macrophage, the microglia, become inflamed. And so in their brain. But one of the things, and so we looked to see if we could recapitulate that. So we took human microglia from iPS cells and made a human organoid from the same patients. And we transplanted the microglia into the organoid in a healthy neurotypical. And we did the microglia into the autism brain as well. And what you see is that you can recapitulate this phagocytic or larger activated cell within the brain. And one of the advantages of this model that we're promoting is that you can dissect out healthy microglia from diseased host. So in this case, what we did was he took ASD neuronal or autism organoids with neurotypical microglia. So these are healthy microglia, and then a normal healthy cortical with, again, healthy microglia. And what we see is that the healthy microglia, as we saw before, when transplanted, they are healthy. They have, active processes, but even the healthy microglia transplanted into the autistic brain result in an activation, a reactivation of the microglia into an inflammatory state. This is a way of saying it's really the host environment that's driving the phenomena, even in a healthy cell. Just a demo of what's going on. Here's another one that it's this new data, unpublished data, with Lisa Mitchell and Simon Schaefer, who used to be a postdoc in the lab, now has his own lab in Munich. So there was this fabulous paper that was published a couple of years ago in Science in mouse, where they showed that if you activate an immune response within a tissue, and this was in a peripheral tissue, and then you look even a long time later, the repercussions of that initial event persist for a very long time. And this means that there's a memory. There's an immunological memory that persists for long periods of time. We wanted to take advantage of our system to do this. So we treat the animals with LPS, activates the microglia, and 1 hour later you see a typical response, even within the organoid. I showed that to you already. But then we look 10 weeks later, and then we look 26 weeks later, and see if we can see any of the changes that are going on. And it's really quite striking. So here's an example of an animal organoid that had been stimulated 10 weeks earlier. Here's the control. And let me just see if I do this whole thing. Yeah. And you can see these differences between these control, and then 10 weeks later, this is an animal that has no stimulation for 10 weeks, no LPS, and they're still blunted. They're still activated. They retain their active state. And we can monitor the size and amount of activity that the cells have. So it's a retained event. when we use our other assay, where we kill the individual cell, we watch the microglia processes as they come in to phagocytize the dying cell. That's the control at 26 weeks, so it's still very active, even in an older animal. But this is what happens 26 weeks earlier. They receive an IPS response, and they are blunted. This response is severely blunted. We've gone on now, and I won't tell a lot more about this, but it's going to be coming out soon, where we have looked at single-cell sequencing and used other omics to identify the molecular change that's occurred in the microglia that's persisting over time. And we're looking at ways in which we can reactivate these cells, either by deleting the microglia and transplanting new healthy cells, which is a very exciting part of the study, or by other methods to reactivate. So I think we have this microglia platform now for long-term differentiation, which is hampered in the in vitro setting. And we think this is a system we can use to study neuroinflammation in the context of the human brain. So let's move a little bit more now to another cell type that we feel is an important role. I can't emphasize enough saying that I think people have we spent as neuroscientists too much time thinking about neurons, and we're not thinking enough about the other cell types that are important. So that's sort of part of this pitch today, is encouraging us to incorporate microglia, astrocytes, endothelial cells, parasites. This is really the nature of these cells that we need to incorporate into our understanding. So we developed a protocol where we can now, if you make organoids and you wait long enough, say 6-10 months, you will begin to see astrocytes forming on the outer side, not throughout, but it's a long-term experiment. So we've developed a protocol, really an amalgamation of things from other people, to enhance the rate at which the astrocytes are formed. And it's really built on an early observation by Lawrence Studer, who identified this molecule of NF1A, which is a switch. And when that gene comes on, the cells initiate the process of becoming an astrocyte. In fact, you can overexpress this in an iPS cell and generate more astrocytes. So here's our organoids that have been stimulated with LIF1, our cocktail for inducing this organoid-induced astrocyte. And you can see already at 21 days, there's already the beginnings of the expression of NF1A. When we look at more markers that you think about as being astrocytic, here's again our in vitro. This is all in vitro at this point. An increase in GFAP already at 60 days. So it's really an enhanced way of being able to explore astrocytes. Doing single-cell sequencing, again, at the in vitro setting, we have a nice collection of mature astrocytes, immature astrocytes, and progenitor cells. We also make, we transduce these iPS cells initially with GFP so we can sort the astrocytes out. We're using AAV GFP reporter. And we take them out at 3 months, and they look at them in vitro, and they're maturing along nicely. But if we take them out at 5 months, they're really quite more mature. So they're progressing through a maturation phase, and by 5 months, they're really quite mature with long processes. They're responding to glutamate stimulation. As evidenced here, this fact about astrocytes is that we think of them as support cells, but they actually are signaling information. Here's multiple different types of iPS cells, and ES cells that we've used to enhance astrocytes and stimulate with glutamate. We get a nice glutamatergic spike out of them. But now we can transplant these cells, and this is where it becomes really quite fun. Here already, by 60 days, you can see that there's lots of GFAP positive cells. So this is 60 days compared to normal healthy human, which takes a much longer time for these astrocytes to come in. And we know that they're astrocytes. We can do the cross-staining and show that they're human. One interesting feature of this is that the density of astrocytes in vitro in vitro organoid. It's really dense for the astrocytes, but the astrocytes in the transplanted cells are significantly less. They're distributed, and I'll go into this. This is work that was done by Mayan Wang in my lab. Initially, it was another postdoc who was helping her at the beginning, but Mayan really carried this over the threshold. And one of the things we can see is that as expected, human astrocytes really look very different than mouse astrocytes. So here's a mouse astrocyte. Many of you have seen these in tissue sections where they're sort of a halo of branches. These astrocytes are really quite different in vitro, or in vivo. And one of the features is you see them attached to vasculature, which is, of course, part of their role. And not only that, but we also see that there's 4 different types of astrocytes that emerge out of these organoids. You'll notice here, just a little bit of a story here, is that the rosettes that you normally see in organoids disappear because the cells have matured so much that now it's just a cortical layer of cells, and the layering is really quite representative. So these are what are called peel astrocytes. These are protoplasmic, and these are myelin or fibrous astrocytes here at the bottom. So they're layered in a nice way. Not only that, have been reported in human postmortem tissue 4 different types of uniquely different astrocytes, including this varicose astrocyte, which is something that are only found in primates. And here we see them in our organoid with these little varicose buds along the way, sitting in the region where the protoplasmic cells are sitting. Going back to this point of the association of the astrocytes with the vasculature, if we take that to an extreme, we can do three-dimensional reconstruction of what we call the vascular unit. And the vascular unit is made up of endothelial cells, pericytes, astrocytes surrounding for the blood-brain barrier. And here we can see that this is the astrocyte. This is the human astrocyte. This is the endothelial cells here. These are the tight junctions. And this is the pericyte, and this is the basement membrane. So within these organoids, we get the full panoply. And these organoids, quite remarkably, form a blood-brain barrier. So here's when you inject a dye into the orbital fissure of the eye, you end up getting blood, of course, going to many areas of the brain or body. So there's no blood-brain barrier in the heart or liver or kidney, but in the brain, it's really quite nice. And actually, the density of the blood-brain barrier within the transplant compared to the contralateral cortex is pretty similar. So they are not only forming blood-brain barrier, but it's sufficient to function appropriately. Doing single-cell sequencing on this to identify the maturation that occurs. It's what's great, of course, now with all those data sets that are out there, we can go into all the human data that's been published and match it together against our data that's generated through from the organoids. So here we can take in vitro 5 months and then in vivo 5 months and compare that to 5, 6, 7, and we look at how they mature. And what's really quite fun is that if you look at this 8-month time point, you have these GO terms. And using gene network analysis for the astrocytes, we see really mature forms. So they are maturing really quite well. Mayan went and took it one step further and did spatial transcriptomics profiling using GeoMax. And so this way, what you're doing is you're getting a ROI, and she counterstained with SOX9, which is a marker for astrocytes. And then she can look in each one of those cells and see what are the genes that are being represented. And she cleverly looked at the peel versus protoplasmic versus the subcortical fibrous astrocytes, and using SOX9 and able to then use that for the segmentation. And what we do is what she sees is a really nice separation between the white matter, protoplasmic, and the peel cells. When you go into it even a little more in depth, you can look at the genes that are expressed uniquely. So here you're comparing cortex astrocytes from peel astrocytes or white matter astrocytes from cortical astrocytes, and they're really uniquely different. So location matters, and who you are and who you become is really dependent upon where you are. and even if you're astrocyte. And so I think part of the takeaway here is that it's not just that you're generating astrocytes, that you're generating the appropriate astrocytes for the location where they are. And a feature I think we need to keep in mind, and we're pursuing this whole idea of location-dependent specificity of differentiation and function. Now, the astrocytes themselves are maturing, but for the neurobiologists in the audience, we also know that astrocytes can play a role in getting neurons to mature. They're helpful. So here we're looking at a modular score of neuronal features, and we're just looking at the neurons now in the single-cell sequencing, and we can see that they are, oops, I just gave away the hint there. There. So in the in vitro setting, they're mature, they're neurons, for sure, but the maturation state or similar score for maturation is really highly increased. For the aficionados in the Alzheimer world, there's a gene called tau, which is expressed in a fetal form or prenatal form, and then postnatally it forms, has splice variants that only come up postnatally, and that's the form of tau, which is also involved in Alzheimer's disease. But what we see is now in the in vivo setting, we're getting this late onset of this mature form of tau. So not only are the neurons surviving and doing well, but they're maturing and differentiating into a much more mature state. We can see this at the electron microscopic level, where we have both pre- and postsynaptic densities being formed within the organoid. Okay, next steps. I'm going to tell you some new things. So we want to, and what Mayan's working on now is trying to get human endothelial cells and parasites inside the organoid, so we have a humanized vascular system. Another cell missing in these organoids is oligodendrocytes, which are the cells that wrap around the axons. Those are missing from our disease, and of course we're doing disease modeling with what we have in each one of these stages. And we're also interested in using these to understand evolution. So we have iPS cells from chimpanzees, bonobos, and I'm not able to talk to you about that, but it's a really interesting story related to astrocytes. But just as a teaser, so here's an 8-month-old organoid, and we do see, this is for those that are familiar with this, this is a myelin sheath around an axon. So I'm not saying that this is happening on a regular basis, but it's permissive for myelination to occur. So we are really optimistic. Okay. So what I've talked to you about mostly at this point is iPS-derived neurons. And one of the issues with iPS-derived neurons is that when you take a fibroblast from an elderly individual and make iPS cells out of them, you've really converted them back into an embryonic state. They're embryos. So if you want to look at aging, or you want to look at age-related diseases in your model systems, you're missing out a little bit, because they're never going to get to be 85 years old, at least, not many of them. So aging is a really important aspect of many diseases that I'm particularly interested in, but heart disease, various influenzas, of course, age-related decline, and Parkinson's and Alzheimer's disease are all there. So when Jerome Mertens was in the lab, he and others in the group developed, were trying to model for Alzheimer's disease, and what they found was, okay, let's take aging and see whether or not we can capture aging with these cells. We took skin biopsies from people from 1 week old, or subject 1 week old, to 80 years old. You can differentiate them into all different kinds of cells, including neurons. And then we did RNA-seq on these neurons from this whole range of individuals. We really found no differences between 1 day old and 80 years old, even though they're neurons, and they're from people that are old. So Marius Wernig had been doing this in mouse and showing that you could use NGN2, for example, to drive a fibroblast into a neuron. You combine that together with ASCII1 and a variety of small molecules that Joe Hurdy in the lab developed that enhance this process. And then you can differentiate the cells directly from a fibroblast into a neuron. They look really quite remarkable. And what we do is, so here's the fibroblasts in a dish, and they are stably transfected with these two genes, NGN2 and ASCII1, under the control of doxycycline. So they're stably transfected. We grow the fibroblasts up to a confluent state. We hit them with dox, and I'm just going to look at and say, I don't know how well you can see this, but they're, put your eye on one cell and just watch it convert from a fibroblast into a neuron. It's really quite striking, and by 21 days, to see them becoming crystal. So here they are. That's they're TUJ1 positive. So you had transformed a fibroblast, a skin fibroblast, into a neuron. Works really well. And now we can do this with aged individuals, and we can identify genes that are unique to the individual from whom you took the biopsy. And I'll just say at this point that we continue to use this. We've made these vectors available in AdGene, and many publications have come out of this network. What I want to tell you about right now is a dilemma that we've had all along. And I put this together with the work that we're doing with organoids, because all the INs right now is a two-dimensional state. And we would really like to work this into a three-dimensional state. So Sam Asalami came to the lab several years ago, and I challenged him with this. I said, can you figure out how to do this? And after a lot of trial and error, he came with this very clever methodology where we take our existing fibroblasts that are stably transfected and doesn't convert them. He allows them to form a three-dimensional matrix of fibroblasts. And then he turns on dox. And when you do that, the cells inside these fibroblasts convert in a three-dimensional matrix into what we call now a spheroid. And here's an example of what they look like. And the beauty of this is he takes 100,000 cells in each of 96 well plates. So you have 10 million cells in a well, and he can just put them through. And then you have, as I'll show you, some pre-standardized sets. We can also do this, I'm going to come to this a little bit later, with astrocytes. So here's an example of what this looks like. This is stained for TJ1, and here's a sort of three-dimensional view through that. You can use this with microscopy now, so you don't have to section. You can actually just, because these are actually quite small. They're less than 1 millimeter apart, so you don't have a core forming in there, and so the flow of nutrients will get through. We can put these on these Maxwell EFIS plates, and quite remarkably, they are really nicely active. So there's three, you can see there's three different organoids on the plate, and we get the activity measurements there. Another way of showing activity is using calcium imaging, is using these fluorophores that we can incubate the cells into, and you can see that these mature adult neurons in a three-dimensional matrix are connected and spontaneously firing. There's a series of experiments we have underway using these to test them. And really, this is more recent data. We haven't, none of this is yet published, but we did single-cell sequencing on them to really see what other cells in there. And we do see that it's a fraction of the cells are not yet converted, but the majority of cells have converted into mature neurons. And even within the neuronal state, we see a variety of different neurons. So these are clearly astrocytes. So here's the precursors, intermediate, and this is the population of different types of neurons that are existing within this sphere. And I would say, for those that are watching this story unfold around the world using the INs, oftentimes we get about a 60% conversion in vitro, and we have to sort out PSA-NCAM to purify the neurons. Here, the efficiency is so high that we just do single-cell sequencing, and we can identify the cells or not. And the sort of really nice convergent here is we do what's called epigenetic clocks. So using epigenetic clocks, you can identify the methylation state of the cell and determine its, quote, biological age compared to its real age. So here are iPS cells, and they're all 0 years old. So even though they've been in culture for a very, very long period of time. And here's our IN spheroids. Here's a cluster of 4 different people that are around 30 to 40 years old. These are our young controls. Be happy that you're 40 and you're young. And then here's our older guys up here. So not only, so we can capture the age is what I'm trying to say there. And then here is what I sort of hinted at and didn't convert the slide. So we can, in our initial starting well, Sam puts the fibroblasts, but he also can put what we call astrocyte progenitors, a method that we developed early on for making iPS cells into a precursor astrocyte that then together with the neurons differentiate. So now we can add astrocytes to our 3D matrix, and we see these nice astrocytes along with TUJ1. And this slide is just to sort of show you one of the issues about organoids is the variability that occurs. Because we're doing this in these 96 well plates, and they're retaining their size. So these are 8 different subjects, and they are all very much have a lot of similar properties to them. Another recent observation that we have working with Jasmine Ravenna in the lab who's our sort of microglia queen. We have put those into this organoid setting as well, and the microglia also are surviving and maturing. I'm not showing details of that right now, but they're still underway. So we're really trying to put more mature cells in here. See, okay. And the final bit is I talked all about this transplantation part and the vascularization of it. And together with Lisa Mitchell, who I talked to you a little bit about before, who is doing the transplantation and treatment with IPS, with the inflammagens to activate the microglia initially and look how long it lasts. She transplants these cells, and she's transplanted the spheroids also into our cavity, and they become nicely vascularized. And then even months later, they are expressing mature markers for MAP2, NUA, and TUJ1. So much like with our organoids that capture the early portions of neural development, in these aged spheroids, we're also have the same sorts of objectives for humanizing with vascular, getting our oligodendrocytes, and we have more data on age-related and evolution with this story too, but it's just I want to give you a flavor for the directions we're going and what we're excited about. And so here are the people. There's Simon, who really did the microglia organoid combination together with Lisa when she was at Tech, and now she's a graduate student in the lab driving this other project. That's Sam, who is the spheroids, and Mayan, and Li Zhang, who initially worked with Mayan to get these astrocyte organoids going. Very fortunate to have a really talented group of people in the lab, great collaborators at UCSD and at the Salk, and we continue to be supported by both private and federal funds, and we're very supportive of that. And with that, I'm done.
We have some time for questions.
Yep.
That was a tour de force as usual, Rusty. So I had a question. So you're doing humanized mouse models with these assembloids. What about putting them into an immunocompromised mouse that makes human stem cell factor IL-3, GM-CSF, or has the inflammatory cytokines that are associated with aging?
You mean you want to transplant the cells into an age model?
No, into an NSG-SGM model. So you can transplant into NSG or into ones that have inflammatory cytokines, so you recapitulate inflammation that's systemic.
No, that's absolutely right. And yes.
Pinar?
Thank you so much, Rusty. That was amazing as usual. I have a question, so maybe I missed, but when you're doing the Ispheroids, which I thought it was really great, and you add microglia, are those iPSC-derived microglia or directly induced microglia? And if so, are you able to make those microglia as well?
Right. So I have to say this is work in progress. So this particular experiment was done with Jasmine, and she has a really good protocol for making iPS-derived microglia. Separately in the lab, we have methods for directly converting fibroblasts or blood cells also into microglia. So we capture the aging. What I showed you here was iPS-derived microglia. We don't have enough data to say about the aged microglia, but that's going to be a real rich story for us. Obviously, is being able to capture aging is a important thing.
Hello, thank you so much for the talk. I was wondering, for the ASD organoids, when you implanted microglia, how was it done, and were they yolk sac derived?
No, the microglia?
Yeah.
No, these are iPS-derived. So we take them from the patient. So we take from the ASD patient or from a neurotypical, and then we take the wild type and put it into the organoid that was made from the ASD, which is mostly neurons. And the point there was that the healthy microglia in the context of ASD neurons became inflamed, suggesting that even if you're healthy, it's the environment that's determining to a large extent your fate.
Right. And
that's not to say that there aren't intrinsic properties involved in this too, but I'm just saying the environment can have an impact, yeah.
Right, and I was just wondering, how much percentage infiltration did you get of the microglia? Were they injected, or were they just...
No, we actually, as they're forming, we mix them together. And so we allow for the fibroblast and the microglia precursors to come together. And then they differentiate. We switch them into differentiation media, and that's when they differentiate. And this is a lot of the magic that Sam's put into it is, how long do you actually differentiate them into precursor microglia before you mix them in with the neurons? Because once you have them together, you have to have a single media. You can't have both media in there. So you got to wait till they're mature enough so they can account for that. Good questions, though, yeah.
Well, Mike Lewis from Cedars-Sinai, very fascinating talk, not my area though. But I was just wondering how advanced disease modeling is, for example, advanced Alzheimer models or Parkinson models, and more importantly, has the field looked into those models with regard to, meaningful therapeutics in order to advance disease treatments for that?
Well, that's the other talk I could have given, with the disease-oriented model. And we've done a lot of work and published actually quite a bit on the INs. So these are the induced neurons, mostly in a monolayer setting. And as I reported and we published, is it captures the age of the individual, but it also captures, we'll just talk about Alzheimer's for a second. It captures some of the disease-related phenotype that we hadn't previously thought much about. So much focus is on amyloid and tau. We're not thinking so much about the underlying biology of the cells. And so we've discovered that there is a metabolic shift that occurs in Alzheimer's disease. If you have age-matched control patients, so 85-year-old controls and 85-year-old diagnosed Alzheimer's disease patients, these cells have a higher proportion of what are called senescent cells. This is something Joe Hurdy discovered and published on. They have a metabolic shift from oxidative phosphorylation into glycolysis. This is in the age-matched condition, but all old, of which you don't see any of this in the iPS-derived, even if they're Alzheimer's-derived. The other sort of fascinating that goes along with this metabolic shift is the neural identity of the cells occurs. And this is something that's occurring. I'm giving you more of an answer than you want to know, but I like this, this whole area of research. The Alzheimer's disease seems to de-differentiate metabolically, as well as in terms of the first glimpses of this now are using biological clocks. We see that the Alzheimer's disease cells look like they're turning back the time. They're de-identifying themselves. So a lot of the mature neuronal markers are decreased in their expression levels. So they're becoming sort of immature. We call this de-differentiation. We're trying to get our head around how to think about this because it's sort of a newer phenomenon for us. So to cut this off a little bit, this mechanism or this methodology that we're developing is useful in disease to look at things that incorporate the age component in the analysis of it. We are now doing these same sort of things using the Ispheroids to see whether or not we can see even additional things in a three-dimensional setting than we have not. But I went too long on that. Good question.
So I noticed one of the markers that you were following, I think was aquaporin 4.
Yeah.
Have you ever tried to take cells from neuromyelitis optica patients or Devick's disease to turn them
No, that's
into
a great.
model?
That's a terrific. I mean, it's clearly a feature of astrocytes, and I realize that this is a disease-related phenomenon as well. But we have not done that.
Okay. Because I think it's turning out that a good percentage of patients that were previously diagnosed with multiple sclerosis,
Right.
in reality have Devick's disease,
Yeah.
which is an aquaporin 4 channel mutation.
Well, what's fun about this is that these astrocytes that are transplanted make these mature markers like that we don't see very much in vitro. So in our immature cells that we can retain and for 8 weeks or so, but if you transplant within the context of the organoid, in the context of blood vessels, that's when we see them coming up.
Here. Thank you. That was fascinating. I wondered, as a vascular biologist, whether you have transplanted aged, let's say, spheroids into young mice and vice versa. So having like a young vasculature, host vasculature, vascularizing your aged or, spheroids and the opposite, because I think that the vasculature also young versus old.
No, you're absolutely right. So the dirty little secrets about these immunocompromised mice is that they don't live as long as you like. So well, it's still 13 months or so. They're pretty healthy, but we're pushing the limits there. The other approach we're taking to this is because, and the other thing I want to remind you is all the vascularization story that I told you here is chimeric. These are mouse endothelial cells. These are mouse microglia cells. So there is a methodology where you can, and you probably know this, that are called vascular organoids. So you can take iPS cells and make really amazing tubes of endothelial cells and parasites. And then you can sort them into different sorts. And Mayan has been doing, who's actually in the audience, she's been doing this for some time, trying to get the human endothelials and parasites in the right percentage and then reorganize them within the host and then transplant them. And with the hope of having sort of an anastomosis of the mouse to the human, and then we would have, but we're not there, looks, yeah. But then we could, anyway, there's lots of things to do. Yeah.
So I think we should stop here, but I have one clarification. So in the early stages that you mentioned, the organoid transplantation, if I understood correctly, you still have the mouse microglia there. And will the mouse microglia penetrate into the organoids, and how do you untangle the human and mouse microglia there?
No, that's really good. Okay, so this is a really wonderful separate story. When we transplant our normal healthy organoid without microglia in there, the microglia from the host migrate in. The astrocytes do too. If you put the microglia in beforehand and transplant them, the mouse microglia stop at the border. They will not go into the human organoid if it contains human microglia.
Sounds like surfing, just locals, right?
Well, but they are secreting chemicals, and so there's either it's a developmental issue, or it's a fundamental species difference recognition. I don't know the answer, but it's something. If I had time and more people, I'd go down that road. It's a really interesting question.
Amazing.
Yeah.
All right, let's thank Rusty again.
Yep.